All right, Bio 201, chapters 2 and 3. This is Bio 111 review, part B. We left off on the last video in covering the basics of solutions and passive transport, diffusion, and osmosis. Review that as you need to understand that as best as you can. And also, don't forget about osmotic pressure and hydrostatic pressure, the two opposing forces in the body that drive a lot of transport of materials. And also the basics of some calculating solutions like percent and moles, and then molarity, and then finally osmolarity. Now on the exam, you're not going to have to do any calculations. You won't need a calculator. However, you will need to understand the concepts of the moles, molarity, and osmolarity, and so forth, and simply take some information and answer whether a solution is hypertonic, hypotonic, or something of that nature. Very similar to what was on pre-lab number 2, however, there are no heavy calculations involved. For example, I could say that there is a given solution of, let's say, any solution that has an osmolarity of 100. And then the question would be, which of the following solutions would be, let's say, isotonic to that particular solution? And then the choices would be given with molarity, perhaps millimolarity, something like that. And then you'd simply have to know the dissociation factor, as I call it, for each one. So if the first choice was, let's say, 100 molar sodium chloride, then that would not be a correct answer because sodium chloride dissociates into two parts, and that would have a 200 osmolarity, whereas the solution in the sample question had 100 osmolarity, and therefore that sodium chloride would be hypertonic to it, not isotonic to it. So it's simple things like that that you can do in your head, or if you need to write on the side of the sheet of paper, that's perfectly fine as well. By the way, 100 and 200 osmolarity is ridiculously concentrated. I don't think that even exists. However, it was just to give you an example. So we're continuing on with finishing up things on transport with different solutions and so forth. And to do that, we can start on page 96 in the purple book. And actually 95, let's start with 95. Give you a second to pause it if you need to get to the page. And we finished the last video with simple passive transport, and we're going now into a slightly more complex types of transport. First one is carrier mediated. So osmosis and diffusion, as we talked about last time, involve just transport of ions, small things, through the plasma membrane, usually through channels or sometimes through the lipid bilayer itself. However, the other forms of transport are going to involve membrane proteins. Remember, membrane proteins have a lot of different functions, and one of those is to transport things from one side to the other. And so carrier-mediated transport is one form of those, and this is simply a mechanism that the cell uses to take something on one side and move it to the other side of the membrane, either out of the cell or inside of the cell. There are two main types of it, co-transport and counter-transport. Self-explanatory. Co-transport is moving things in the same direction. Counter-transport is moving things in the opposite direction. Now on page 96 is the general example of things that involve carrier proteins, and that is facilitated diffusion. Like diffusion, remember, facilitated diffusion also goes along a concentration gradient, and it is a passive process. The only difference is a carrier protein in the membrane helps move the solutes from one side of the membrane to the other. It's no more complicated than that. Then the next large category of transport is active transport, and this is the type that requires energy because we are moving things against the concentration gradient. We are going from low concentration of something to high concentration of something, and that requires energy. Now the key word here is energy. Yes, ATP is often the energy source, but it doesn't always have to be. Think about the electron transport chain in the mitochondria. ATP was not the source of energy for creating the proton gradient. It was the energy dissipated by the electrons going down the transport chain that released the energy to the pump proteins to move the protons from one side to the other and create that gradient. So don't assume that ATP is always the energy source for active transport. It often is, but it doesn't have to be. So that's the simple explanation. Active transport going against the concentration gradient, energy required. Now, there is the I guess if your book calls it primary active transport, yeah, that's on page 97. That's the sodium potassium pump, classic example, always used to illustrate the active transport process, where sodium ions are moved outside of the cell and potassium ions are moved inside the cell. This is another form of counter transport in active process in this particular case. In 111, you probably just had to memorize it for the exam, whatever it was on, and maybe nothing was said about why cells would expend energy creating this artificial gradient of sodium ions on the one side and potassium ions on the other side. When we get to muscle cells, chapter 10 and chapter 12, neurophysiology, you'll see why that's important and why that's useful. But for right now, just know that the thing exists and be familiar with the pump process itself. The other form of active transport is secondary active transport. And this is where there is some form of active transport like the sodium potassium pump, which is going to create this artificial concentration gradient. And as a consequence of that, a second transport process can take place to move substances from one side to the other. So your book has an example of this. It's on page 98, utilizing the sodium potassium pump, and then the sodium ions are going to be part of a second transport process, which will move materials into the cell. There is also an example, I believe your Bio 111 textbooks had it, where a proton concentration is produced, and then the hydrogen ions are used in a secondary transport to bring sucrose into a cell. So that is secondary active transport. And then the last categories are the vesicular transport. And vesicles, which are these membrane-bound spheres inside a cell, are these carrying units which can transport things throughout the cell, and they can also be used to transport things out of the cell. And then if a cell takes something in, vesicles can also form in that process. So when things are transported out of the cell by vesicles, that is exocytosis, and there's really nothing else to that. When a cell takes something in from the outside and a vesicle forms, that is endocytosis. And there are three different types of endocytosis. There is pinocytosis, which is sometimes called cell drinking. It is basically just whatever liquid happens to be outside the cell, water primarily, and whatever ions and maybe small molecules happen to be there are taken in by the cell. In pinocytosis, the plasma membrane essentially invaginates in a little bit, and that almost creates, think of it as a suction that will pull the liquid solution contents into that little valley that's there. Then the membrane closes behind it, and then a vesicle is formed inside the cell and goes wherever it needs to go. The second form is phagocytosis, sometimes called cell eating, and this is where a cell can take in a very large object, a white blood cell taking in a bacterium or a piece of cell debris or something like that. In this case, in phagocytosis, the cell actually reaches out and engulfs something. The pseudopods are produced where the cell reaches out and engulfs something, surrounds it, and then a vesicle forms on the inside. The third and final form is receptor-mediated endocytosis, which is a specialized form of endocytosis where membrane proteins that act as receptors are involved with this. Another function of membrane proteins, communication, receptors being one of those, where whatever substance or molecules are on the outside of the cell have to bind to these receptors, and then only when that happens does the cell take in whatever is out there and cause endocytosis to occur. There is a picture of that on page 99, specifically receptor-mediated endocytosis. In that level of detail, don't worry about it. Just know that it is a highly specific form of, excuse me, endocytosis. Don't worry about transcytosis. That's on the bottom of 98. We're not worried about that. On page 99, down at the bottom, it talks about the membrane potential, the resting membrane potential. I'm going to just say at this point, know what it is. It is the voltage difference between the outside of the cell and the inside of the cell, an electrical difference, sort of like a battery with a positive terminal and a negative terminal. All cells have this membrane potential, this resting transmembrane potential, as it's officially called. And it'll become important when we get to chapter 10 on muscles and also chapter 12 on neurophysiology, because we'll see that a high resting transmembrane potential, such as produced by the sodium-potassium pump, is critical to the functioning of the cells that we're going to be talking about at that point. But for right now, know that it exists. Hopefully it was at least mentioned in Bio 111 without going into too much detail. So it just, again, know it exists, and it is a mechanism that is used by cells to respond to their external environment. Okay? So leave it at that for right now. At this point, on page 100 and 101, there is a double-page thing summarizing the transport processes. Passive transport on the left, active transport on the right. It's a great overview. A lot of the same pictures and things that were in the previous pages are there. So it's a great summary on all of these things that we've just talked about. So that finishes up the transport things. On page 102, on the top left there, it finishes the business on the resting transmembrane potential. So just take a look at that to be familiar with it. We're going to come back to mitosis and the cell cycle later on in this video. Right now, what I want to do is go back to chapter 2 and kind of pick up where we left off from there. So we finished with pH and aqueous solutions, and I'm going to page 45 at this particular point. If you need to pause the video and get to page 45, that's fine. Just turn it on again when you're there. And on page 45, it begins talking about the organic molecules and the biological molecules. So a few pages earlier, it talked about organic versus inorganic. And just to review real quick, organic molecules are carbon-based and with hydrogen associated with them. We're going to use that as our standard definition. There are a few exceptions to that, like carbon tetrachloride, but carbon dioxide, carbon monoxide, those are not organic molecules. For our purposes, an organic molecule is carbon-based, and then hydrogens associated with that in the covalent bonds that are going to be the majority of the molecule, hence the name hydrocarbon. And all biological molecules, the ones that we're going to be dealing with, are going to be based on organic molecules. Now, not every organic molecule is good for you, but the ones that we're dealing with in living organisms are the ones that are good for you for the most part. So before we get to that, the bottom of page 45 has a table there with functional groups. Excuse me. And you learned about functional groups in 111. Now, this table only has 4 of them here, and in 111 there were several others, and we need to be familiar with all of them. So I'm going to go through them real quick. And for functional groups, you don't need to know anything super complicated about them. Know their name, obviously. Know the molecular formula for those, like OH is the hydroxyl group, and then know the properties. of the functional group, whether it's polar or nonpolar, whether it's acidic or alkaline, and in the case of the sulfhydryl group, that it can form disulfide bridges. So that's the extent that I did it in 111, and then we don't have to go any further than that. But you do need to be familiar with those. So 4 of these are in your book, as I mentioned, and then 4, the rest of them, 3 or 4 are going to be in the Bio 111 book if you still have it. If you don't have that, then I'll mention these real quick so you have them as well. So I'm going to do it in a slightly different order. The hydroxyl group OH, or OH minus technically as an ion, is probably the simplest one. It is a polar functional group because of the negative charge. Anything that has an oxygen is going to have polarity associated with it. I mean, that's a given. If you know that and can remember that, whenever you see a molecule with an oxygen anywhere in it, there's going to be polarity involved in some way. So a hydroxyl group, OH minus, has polarity as its property. So if it is attached to some organic molecule, if it's a very small molecule, then the entire molecule will become more polar. If it's a very big complex molecule, then the place where the OH is actually attached will have some polarity to it, and the rest of the molecule can be whatever, depending on what the components are. The next one, and this is one that's not on that list there, is the carbonyl group. That's the C double bond O. Since there's an oxygen, again, we have polarity. And there are 2 subcategories of the carbonyl group. There is the aldehyde and the ketone. The aldehyde is when the C double bond O is on the terminal carbon of a particular molecule. The ketone is when the C double bond O is somewhere other than the terminal carbon. So real quick, I can draw this on the board. So this represents a 3-carbon molecule, and the double bond O is on the terminal carbon. This is an aldehyde, whatever the rest of the molecule happens to be. If, on the other hand, we have something like this, and the C double bond O is anywhere other than the terminal carbon, that is a ketone. And again, because the oxygen is here, polarity. Little hint for those of you that didn't have me for 111, and maybe those of you who did and forgot. Most of these functional groups are going to have polarity as a property. There's only one that doesn't, and we'll get to that in a little bit. So we did the hydroxyl group, OH minus, the carbonyl group, C double bond O. Next is the carboxyl group, which is C2 oxygens and a hydrogen. And that one is in your book. There's the second one down. And the carboxyl group, now with 2 oxygens, obviously lots of polarity, but also acidic properties. The carboxyl group makes things known as carboxylic acids, which are found in various places in cells and so forth throughout the body. Then we have the amino group, which is NH2. And when you have a nitrogen present in a functional group, that also is going to give polarity, since nitrogen is very near oxygen on the periodic table. It's right next to it. So there's going to be a certain degree of electronegativity, hence polarity will be there as well. However, the amino group also has alkaline properties. Now recall the subunit of proteins, amino acids. That name represents the functional groups that are on that particular molecule, whether it's a very simple amino acid or whether it's a complex one with a big giant ring side chain and so forth. Amino acid still defines the functional groups that are on that molecule, the NH2 on the one side and the COOH on the other side. So that is the amino group. Another one is the sulfhydryl group, SH. Sulfur, which is on the row below oxygen and nitrogen, also has a certain amount of electronegativity, so polarity, again, is part of that. But the sulfhydryl group has a unique thing where you can form what's called disulfide bridges between 2 of these if they come into close proximity to each other. Recall your tertiary protein structure where sulfhydryl groups, if there are 2 amino acids with sulfhydryl groups coming near each other, a bond can form between them. That's the disulfide bridge that helps hold the protein structure together. Another one is the phosphate group, and that one is in your book there as well. Phosphate, POO3, there are 3 oxygens there, heavy duty polarity. Where do you find phosphate groups? In the phospholipid bilayer structure of the plasma membrane. Remember, the polar heads are the phosphate groups, the fatty acid tails are the nonpolar areas, and when they are opposite each other like this, you get that nonpolar barrier between the aqueous environments on either side of the plasma membrane. Finally, the methyl group, CH3. This is the only nonpolar functional group that we are going to be concerned with. Carbon and then 3 hydrogens around it. It's the derivative of methane, hence the name methyl group, but it is nonpolar. So if a methyl group is attached anywhere to a molecule, it will have nonpolar properties or qualities at that particular point. So that covers the functional groups. And as I mentioned, if you have your Bio 111 text, there's a whole page in there which covers all of those. Otherwise, the 4 of them are here, and then the others that I mentioned, just be familiar with those. Recognize their molecular formula and their properties that each one of those have. Now, with that, we're going to jump off into the biological molecules. And this starts on page 46. And the biological molecules are your food groups, for lack of a better term, your carbohydrates, lipids, proteins, and nucleic acids. And we're going to go through these quick, and any further details to refresh whatever you need to refresh on should be in your Bio 111 notes, and you can also find things in the text in these pages here in Chapter 2. So for carbohydrates, first thing, function. Primary function of carbohydrates is an energy source. These are energy molecules, and when we perform cellular respiration, which we'll talk about later, carbohydrates are one of the primary sources for generating ATP. So they are basically a fuel source, if you will. The other thing carbohydrates do, they have some non-energy purposes. Cellulose, which is more of a plant thing, which we're not worried about here so much in human biology, but that is a non-energy form of carbohydrates. Also, short-term energy storage. Starches and glycogen. Starches, again, are plants. Glycogen is what we use as animals to store for short-term glucose, which is a monosaccharide, and other carbohydrate forms for later use. The generic formula for carbohydrates is CHO, and there's a 1 to 2 to 1 ratio of these molecules in respect to each other. The basic subunit of carbohydrates are monosaccharides, mono being 1, saccharide meaning sugar, so a single sugar. Put 2 monosaccharides together, you have a disaccharide, 3 of them a trisaccharide. Anything more than that we can call a polysaccharide. Monosaccharides and disaccharides are polar, they do dissolve in water. Polysaccharides are non-polar. Starches like potato starch, cornstarch, and things like that don't dissolve very well in water. Anybody who's tried to make gravies is familiar with that. So on page 46, they do have the basic pictures there of the monosaccharides or the carbohydrates there. Also the dehydration synthesis reaction to produce them and the hydrolysis reaction, excuse me, to break them down. And again, a reminder, no matter which of these biological molecules we're dealing with, dehydration reactions produce the larger ones, hydrolysis reactions break them down into their subunits. That's the same no matter which molecule, which biological molecule we're dealing with. So the real quick, some of you may have this already down. If you know the molecular weights of monosaccharides and disaccharides, that's great. Monosaccharides, the molecular weight is 180. Disaccharide, the molecular weight is 342. Think about why. 2 monosaccharides together would add up to 360. However, it's a dehydration reaction that joins them. You are removing a water molecule that has a molecular weight of 18. So you have to subtract that from the 360, giving you the 342 total for that. So those are monosaccharides, and then starches and we're going to focus on glycogen primarily, since it is part of human biology, is simply a long chain of monosaccharides joined together with varying degrees of branching in the molecule itself. And they have that on page 47, the molecular structure that is there, just a whole bunch of monosaccharides together. Don't worry about the specific name of the linkage there. It's a glycosidic linkage, but I'm not going to test you on that. Down on the bottom of 47, there is a table which summarizes the functions of carbohydrates in the body, a kind of review. You can go ahead and look at that there as well. On page 48, talks about lipids. Lipids are, by the way, carbohydrates are polymers. Okay, so we'll get that out of the way before I forget. Lipids, functionally, lipids also have an energy purpose. They're a long-term energy storage. So carbohydrates and lipids, their primary functions have to do with energy, and lipids are a long-term energy storage function. In addition to that, lipids also have some non-energy functions. Phospholipids, for example, plasma membrane structure. And within the body, also, lipids are used as cushioning. They are used as thermal insulation in various places of the body. So there are some other functions as well that these lipids can do. So lipids encompass several different kinds of molecules, but their unifying thing is non-polarity. Lipids are non-polar. They are hydrophobic. And we'll talk about the basic ones here real quick. The most common ones are the triglycerides. And the triglycerides, these are the neutral fats, as they're often called. And these are composed of 4 subunits, a glycerol molecule and 3 fatty acids when they are all joined together. Now, lipids, triglycerides, are not polymers. Once you join those 4 together, that's it, and there is no string or chain of these things to make it any bigger. So, don't worry about the name of the bond that is formed or anything else. Glycerol and 3 fatty acids joined together will make the triglyceride the lipid. And you've heard of saturated and unsaturated fats. That's referring to these triglycerides, and specifically to the fatty acids themselves. Saturated fat is one where the fatty acids are pretty much all straight lines. They are all single bonds. Now, a fatty acid is a long chain of carbons held together with, in this case, single bonds in a saturated fat. And saturated fats are solids at room temperature because as they cool, the fatty acids can all kind of compress and sandwich together very nicely, helping it to solidify at a higher temperature. Unsaturated fats are those which have double bonds in the fatty acid chains, at least one double bond, it could be more. And as a consequence of that, the double bond is going to change the shape of the fatty acid. It is now not a straight chain. It can kink and bend and twist and that kind of a thing. And when that happens, they don't sandwich together very nicely. They have to get a lot cooler before they could potentially solidify. And some unsaturated fats really never solidify. They become more of a slush than anything else. Saturated fats are always from animal sources. Unsaturated fats are from plant sources. Unsaturated fats are the oils that you cook with or whatever. So that is the difference between those. That is mentioned all on page 48. Down on the bottom of 48 and into 49, it mentions another type of lipid molecule known as an eicosanoid. You may or may not have heard about this in 111. It is based on a different molecule, arachidonic acid, but there are different subcategories of these eicosanoids which are important in human physiology. Prostaglandins, which you may have heard the word, are of this example. They're involved in inflammation and do a whole bunch of other things throughout the body, and we'll mention those periodically as we go through. In the immune system, there are certain others which are in this category as well. That's more for 202 when we get to that point. Then, if you flip over to page 50, on the left-hand side, there it talks about the triglyceride formation. Again, dehydration forms it, hydrolysis breaks it down. No more complicated than that. On the right side of 50, it talks about the steroids. And then there are the examples of those, of some structural formulas on the bottom right. Steroids are also lipids because they are nonpolar. However, they are based on cholesterol. Cholesterol is that big ring structure with that one chain coming off the top there that you can see. And by substituting chains and functional groups and things like that on the cholesterol molecule, you form the various other steroid type of molecules. The ones that most people are familiar with are the steroid hormones that we have. And they give two examples down there, estrogen and testosterone. Estrogen is the female sex hormone, one of them. Testosterone is the male sex hormone. And if you look at the comparison between the two, there is very little difference between them as far as where functional groups are located and whatnot. And just those slight differences make all the difference between physiologically female and physiologically male. And again, that's again part of the lipid category. Then your book mentions on the next page 51 phospholipids. As I mentioned, in the plasma membrane, phospholipids are part of the membrane structure. A glycolipid is simply a lipid that has a carbohydrate molecule of some sort attached to it. On the bottom of page 51 is another table representing the various lipids and summarizing what they do in the body, so you can take a look at that as a good summary. Then proteins begin. On page 52, there also is some more pictures of phospholipids, again for plasma membrane. You can look at that. Proteins are going to continue on with page 53. Now, proteins are the most abundant and the most diverse group of biological molecules that we have because they do so many things. Primarily, though, function-wise, proteins primarily are not concerned with energy. Can proteins be used for energy? Certainly. In regular turnover, when they're broken down and replaced, a lot of times the cells will generate some ATP from proteins, from the amino acids, but that is not the norm. In the body, the norm is for the body to use carbohydrates and lipids. Proteins are used for almost everything else. They are basically non-energy functions of these molecules. And on page 52 and into 53 and even previously, there's a bunch of bullet point things there that illustrate the functions of proteins. You can go ahead and take a look at those. Proteins are involved in movement. Proteins are involved in cellular and body movement, in transport. Carrier proteins carry things around the body in the blood. Membrane proteins have a myriad of functions within the plasma membrane itself. Proteins are involved in cell division, in mitosis, the spindle network pulling the chromosomes apart. And enzymes is a big thing as far as proteins go. So there's a huge array of things that proteins do. Structurally, proteins are composed of amino acids. Those are the basic subunits of proteins, and linking amino acids together form peptide bonds. That's the only bond you really need to be familiar with, the peptide bond. And linking amino acids together in peptide bonds form polypeptides. And polypeptides can be small, they can be long, and technically speaking, the difference between a polypeptide, or just a peptide is sometimes used, and a protein is that the protein is functionally active, whereas the peptide or polypeptide may not be. Again, there are some gray areas. I wouldn't worry too much about the technical definition. There's not going to be a question like that on the exam. But in case it comes up in your reading or you're having a discussion with somebody, that's kind of what that boils down to. So the protein structure is on page 53, the amino acids, the peptide bond, and all of that. Notice it mentions there again the amino group and the carboxyl group, the functional groups we talked about before. And then on page 54, it goes into the different levels of structure. In the Bio 111 book, this covered 2 pages. Here, because it's review, we're condensing it down into one. The primary sequence is, of course, just the string, the chain of amino acids in the order that is dictated that they be in. Think about what determines that order of the amino acid sequence. It's determined by the genes. Okay, the codons coding for the specific amino acid and the sequence that they're in the gene determines the amino acid sequence, determines the primary structure of the protein. That is just the peptide bond linked amino acids in sequence, however long that may be. As a consequence of that, this chain now begins folding, and that determines the secondary structure. The secondary structure, there are 2 types. There is the alpha helix, which is just a coil, and there's the beta pleated sheet, like a pleated skirt or drapes or an accordion or something like that. And what holds that together is largely hydrogen bonds, where we have a hydrogen coming near a nitrogen, perhaps from an amino group, or an oxygen from a carboxyl group, or something on a side chain. It varies, but the hydrogen bonds are going to be holding the secondary structure together. Then we'll continue folding into the tertiary structure, and the tertiary structure is held together by a number of different things. Hydrogen bonds, certainly. Disulfide bridges, we mentioned, if 2 sulfhydryl groups come close together. Hydrophilic and hydrophobic interactions. If we have side chains, the R groups, which are going to be very important in tertiary structure. Remember, R groups are not functional groups. R groups are usually more complex. If we have a bunch of hydrophobic nonpolar R groups all coming clear near each other because of the folding, they will form an attractive force that will help hold that part of the molecule together. Same thing with hydrophilic R groups. If they're polar, will also tend to coalesce together and help hold the protein together. Ionic bonds can also potentially form in tertiary structure as well. And that will yield a completed protein. If there are more things that have to be done to activate it, and when it is, that yields a complete functional protein. Often, though, we can have much larger molecules, much larger proteins made up of individual subunits of tertiary structure proteins, and that leads to quaternary structure. Hemoglobin is often used as an example of this. Antibodies are also an example. There are others as well. And that is the multiple subunits which are completed at tertiary structure now interact together to do a common job. In the case of hemoglobin, it's to carry oxygen, and that's, I think your book is using that example there as well. That's the common one. Completed proteins come in 2 basic categories. There are globular proteins, hemoglobin is one, which basically look like globs, have no real definitive shape. And the second category are fibrous proteins. These things look like threads or strings or cords. And those have the definite shape. And when we get to the muscle chapter 10, the contractile elements that help muscle contract are fibrous proteins. In connective tissue, we'll talk about other fibrous proteins there as well. But those are the 2 main types to be familiar with at this particular point. So the pictures there kind of show very nicely the different stages of structure. It's described on page 55. Enzyme function is mentioned. We talked about enzymes already. On the top of page 56, it goes through the enzyme function, the substrates, enzyme-substrate complex, the reaction takes place, products are formed, they're released, enzyme is back to the way it was. Very simple. Now, proteins, a couple of functional things about those. Protein, and I think we mentioned this in the last video, proteins operate at optimal temperatures and pH. If we go too far away from that optimal range in either direction, then proteins, if they're enzymes, are especially going to lose their function. And when we put proteins into adverse environmental conditions, let's put it that way, there is always the probability that they can denature, which means literally they fall apart. And that is a disruption of secondary, tertiary, and quaternary structure. Denaturation does not affect the primary sequence. Denaturation does not affect the peptide bonds that form. So if a protein is denatured under the right conditions, it can be renatured. It can reassume the shape that it needs to assume. And that is mentioned in your book here as well. At one point, temperature and pH is on page 57. Glycoproteins, and don't worry about proteoglycans, but a glycoprotein is a protein with a carbohydrate molecule of some sort attached to it. And these are typically membrane receptor proteins when we talk about membrane proteins. All right, on 57, we also are looking at nucleic acids. And nucleic acids are found in the nucleus, hence their name, and they're also acidic, hence the name nucleic acids. Function of nucleic acids is information storage. This is the DNA and the RNA molecules that we are generally familiar with on the chromosomes and so forth. So the nucleic acids, there's proteins, again, are polymers, and nucleic acids are also polymers. So the only non-polymer biological molecules we're dealing with are the lipids. So nucleic acids, their basic subunits are nucleotides. Nucleotides are composed of 3 individual subunits. There is the pentose sugar, either ribose or deoxyribose, depending on which one we're dealing with, DNA or RNA. Phosphate groups, PO4, and finally the nitrogenous base. And the name gives you an idea of what's involved. Nitrogen is involved in there, base, obviously alkaline. Okay, so you can deduce a lot of qualities about the molecule by the name. The sugars and the phosphates, when the nucleic acids are formed, form the polymer itself. So the linkage is sugar, phosphate, sugar, phosphate, sugar, phosphate in a big long chain. The nitrogenous bases are off at 90 degrees to that. And so those nitrogenous bases are going to form, when we get to DNA structure, the rungs, if you will. So I just want to introduce that real quick. You don't have to worry about purine and pyrimidine in terms of the individual nucleotides. If you know it, great. I'm not going to worry about that detail. You should know the 4 and 5 nucleotides that are involved in both DNA and RNA. That is shown on page 58. Even though separated by purine and pyrimidine, you don't have to worry about that. On the left-hand side are the names of the nitrogenous bases that also give name to the nucleotide themselves. So when we are looking at DNA, the D is for deoxyribose, that's the pentose sugar that's involved in it. DNA is normally double-stranded, and they have a picture of that on the right-hand side there of page 58. And the double-strand thing is twisted, giving it a helical shape, hence the name double helix. So when we are looking at a DNA molecule, the strands that are on either side running parallel to each other like this, those are the rails of a ladder. If you want to think of the DNA molecule as a ladder, those would be the rails, what you grab onto as you climb up. The nitrogenous bases at 90 degrees pointing toward each other make the rungs of the ladder, what you step on as you climb. Now, the nitrogenous bases of each strand are joined together in the center by hydrogen bonds. And the bases pair up in a very specific way known as complementary base pairing. And in DNA, the 4 nitrogenous bases that we are dealing with are adenine, thymine, guanine, and cytosine. And I said it in that order specifically. Adenine and thymine always pair together. Guanine and cytosine always pair together. That's the complementary base pairing nature of the nucleic acids. And it is hydrogen bonds that hold the two strands together. So the sides, the rails, are sugar phosphate, sugar phosphate, sugar phosphate. The rungs, the nitrogenous bases, are held together by the hydrogen bonds. Excuse me. And the two chains on either side, the two strands, their direction is known as anti-parallel. And I'm going to draw on the board here just a second so you can see the importance of this. So DNA, double-stranded deoxyribose. The D does not mean for double-stranded, it stands for deoxyribose, which is the pentose sugar. And AT pair together, C and G pair together. The importance of complementary base pairing is accuracy in two things: DNA replication and transcription. Okay, things you should be familiar with since you've had this in Bio 111. So that ensures accurate copying, essentially, is what we're dealing with here. Now, RNA is the other nucleic acid that we deal with. RNA is normally single-stranded. RNA, the R is for ribose, the pentose sugar, and RNA, the strand is the same thing, sugar phosphate, and the nitrogenous bases, three of them are the same as in DNA, but one is different. So we have cytosine and guanine, that's the same as in DNA, and they pair together. But adenine in RNA pairs with uracil. That is the other nitrogenous base that we are dealing with. So in RNA, if there's any pairing going on, it is adenine and uracil, A and U, whereas A and T, thymine, is what's found in DNA. So very quickly, let me just put something up here regarding the strands themselves. So what I have up here are two strands of DNA, and I have them labeled 5' to 3', and the little tick marks I'm putting here represent the nitrogenous bases, the base pairing going toward each other. Now the 5' and 3' are important because that represents two things. It represents the direction of synthesis. When we talked about, or when you did Bio 111 and did DNA replication as well as transcription and even translation, 5' and 3' hopefully were mentioned because that is the direction of synthesis of the nucleic acids. When DNA replication takes place, the new DNA strand is synthesized 5' to 3'. When messenger RNA is produced in transcription, the messenger RNA is synthesized 5' to 3'. So that is the obligatory direction of construction, the direction of synthesis of any new nucleic acid strands that are produced. The second thing, as far as 5' and 3' is concerned, it is the reading frame. In the genetic code, yeah, there was the box and it had the codons and everything else, and the codons are all together in what we call the gene or the allele on a chromosome. But the reading frame, to make sense of the DNA language, the reading frame is 5' to 3'. So 5' represents essentially the capital letter, the beginning of the sentence, and the 3' represents the end of the sentence, the punctuation mark, whatever would be there. So those are the two things to be familiar with on that. So that is the basic overview of nucleic acids. Pretty good summary pictures and everything there on page 58. On page 59, up at the top, is a table comparing DNA and RNA. It's a great summary thing to look at. And also on page 59, it covers the last of these biological molecules we're looking at, and that is ATP. ATP, you know, is the energy molecule, the energy currency, whatever derivative of the word is used. The cell uses ATP for all of its molecular, or nearly all of its molecular metabolic processes. And ATP is a nucleotide, kind of like the ones we've just been talking about, but it is unique in the sense that it has these 3 phosphate groups attached to it. And when you have 3 phosphate groups all next to each other, you have remember, phosphates are PO3, and you have all these oxygens, you have all this negative charge. And when you try to put negative onto negative, it's like taking two magnets of the same pole, let's say two north poles, and pushing them together. They will repel each other. But yet, the cellular machinery is able to link these phosphate groups together. So if you look at the picture that is on page 59, on the right-hand side, you can look at either the top one or the bottom one, it doesn't matter. The important thing to get out of this is notice what is between the phosphate groups. The phosphate groups are represented by these yellowish colored spheres. Now, when you talk about, or when you indicate, that's the word, a chemical bond between two atoms, usually it's a straight line. And however, notice what's there. There is a red wavy line between the phosphate groups. That red wavy line means that bond is highly unstable. It took a lot of energy to get that together, and it's kind of barely holding on. Now, the first two phosphate groups, phosphate 1 and phosphate 2, a lot of negative, more negative oxygen, putting that together took a lot of oomph, and then it's kind of barely holding on. To attach the third phosphate group, you got the two of them, all these oxygens, big giant negative thing, and now you want to add another negative thing to it. Even more energy was required. So that second bond that is there is even more trigger sensitive than the first one is. So the ATP molecule, the usefulness of it as an energy source is primarily because of these highly unstable phosphate to phosphate group bonds that exist that can be very easily broken, and then the energy released from that. And the phosphate group is transferred. That's phosphorylation, which hopefully you should be familiar to you as well. So the important thing to remember is that in ATP, there are two usable phosphate group bonds that are available. They are both highly unstable, high energy bonds that can be easily broken. The one between phosphates 2 and 3 is more easily broken because it was harder to make. The one between phosphates 1 and 2 also can be easily broken. It's a little more stable, but it's still a lot of negative, anti-negative there trying to push the thing apart. A lot of students come out of Bio 111 thinking that ATP only has one usable bond there that can be broken for energy. That's not true. It has both of them, and I'm glad this book indicated that both of those bonds between numbers 1 and 2 and between 2 and 3 are usable to tap into for energy purposes. So that's the thing to get away from this particular thing here about the ATP molecule. I'm not worried about the details about the sugar group and the nitrogen and all of that stuff. It's the bond between the phosphate groups that I want to make sure you understand that both are available for energy purposes. So that covers the review material in Chapter 2. And we already did some of the stuff in Chapter 3 as far as the solutions and the transport and everything is concerned. So the only thing remaining is cell structure and cell and organelle function. We'll do that relatively quick. I'm going to spend a little bit of time on glycolysis and aerobic respiration because that tends to give people some trouble. And then genetics, a little bit on mitosis and meiosis, and the cell cycle, and that I believe should about take care of things unless there's something else I missed, and we'll get to that once we do that. So I'm going to stop this video here. Part B will end here, and I'm going to have a Part C, which will cover everything else that remains in Chapter 3.